This method can help answer key questions in the field of protein-protein interactions by enabling the detection of novel binding partners for extracellular proteins, both in high throughput and with high sensitivity. The main advantage of this method is that with just a few micrograms of protein, you can generate hundreds of slides using the microarray printer.
Use a standard microarrayer for slide printing. This instrument has a capacity of 57 slides per run, uses a print head holding 48 spotting pins, and can accommodate up to 8,000 spots per slide. Generate working plates using 384 well plates at 10 microliters of sample per well from the stock plates. Perform this step manually prior to slide printing using the microarrayer. Then, spot proteins with quill-type spotting pins onto epoxy-silane slides at 60% humidity to prevent dehydration of the protein spots. Cy3 labeled bovine serum albumin can be spotted in duplicate between each protein sample to visualize the array for mask fitting. Subsequent to printing, remove the protein microarray slides from the humidified environment. Next, use an ultrasonic fogger to generate a fine mist of blocking solution that settles onto the slide surface. Then, block the slides overnight with 5% milk in PBST to inactivate the surface. Store slides at -20 degrees Celsius in 50% glycerol to prevent freezing.